So, my mic's not working or what? Oh, way turned down. Okay, gives me a lot of background noise. I'm not sure what I'm gonna do about that. We got this thing that's really loud right now for some reason. I could just turn this off. But then we also got. Let me stop the rain. I like it, but it's gotta go. So it's already bad enough just with the sound of my computer here in the background. So let me turn this back on. Actually, I'll turn it down to 23 will work fine. Won't be on now for a bit at least. So one thing I'm kind of scared about with caffeine is this whole stimulant thing. I'm not a huge fan of stimulants. Just not the biggest fan of stimulants. I'm not saying like neurostimulation. Neurostimulation is fine, but I don't know. So, let's start with I thought we already did this, but it's okay. Hey, Arbor, how are you doing?
Hey, I'm doing great. Thanks for asking. I hope you're doing well too. What's on your mind today?
Well, I was thinking about actually some things that have been bothering me in the back of my mind, and it's because I want to know more about the stimulant nature of caffeine and the dopamine system, because it's been kind of scaring me in regards to, and I'm learning recently about the whole thing how dopamine D1 receptors potentiate 5-HT2A receptors, and their function's kind of interdependent. But one thing that I knew before that was that cannabinoid receptor function is also dependent on adenosine A2A receptor function. Is this true?
Absolutely, you're right on point. There's definitely a complex interplay between these systems. For example, adenosine A2A receptors can indeed modulate the function of cannabinoid receptors and vice versa. This interaction can influence various signaling pathways, including those involving dopamine and glutamate. And you're also correct that dopamine D1 receptors can enhance certain effects of 5-HT2A receptors, which is really fascinating in terms of how psychedelics and stimulants interact with the brain. It's a complex dance between these neurotransmitter systems, and it definitely can get a bit overwhelming, but super interesting to explore. If you have more questions or want to dive deeper into any specific aspect, just let me know.
Yeah, absolutely. So I guess one thing I want to start with is the clearing up of something that I've heard thrown around a little bit. And it's the idea that, and I want you to fact check me on this, and I say me because I've heard the facts from someone else. In my opinion, all facts anyone knows were heard from someone else, but that's a philosophical thing. So I want you to fact check me in the sense I want you to sanity check this thing that I heard, which is the idea that dopamine D1 receptors are mainly upregulated by caffeine in the hippocampus, along with the adenosine A1 receptors. So I suppose the A1 and D1 heteromer is the thing that's actually causing the D1 to become upregulated in the hippocampus, and that caffeine mainly upregulates dopamine D2 along with A2A in the striatum in the same way. Is it true that these are the main two brain areas where we'll see dopamine and adenosine receptor upregulation? How much is this the case? Would we see it in other brain regions, and how much more would one be than the other, talking about A2A and D2 versus A1 and D1? Do you think you could give me a guess or a guesstimate as to which way it might go? Would there be more of the D1A1 in the hippocampus, or do you think over chronic use there might be more D2A2A in the striatum instead?
That's a really interesting question. There's definitely a lot of nuanced interaction between these receptors and brain regions. Let me break it down. D1 and A1 receptors in the hippocampus. D1 receptors in the hippocampus are indeed important for cognitive processes like memory and learning. A1 receptors are known to have a modulatory effect on neurotransmission, and caffeine and adenosine receptor antagonists can indeed reduce A1 receptor activity. This can lead to a sort of upregulation of D1 receptor signaling in the hippocampus because you're reducing the inhibitory adenosine tone. So it's true that caffeine can influence the D1A1 interaction in the hippocampus, potentially boosting D1 signaling indirectly. D2 and A2A receptors in the striatum. D2 receptors in the striatum are crucial for modulating motor control and reward pathways. A2A receptors are heavily expressed in the striatum. and they interact with D2 receptors, often in a way that can modulate D2 receptor signaling. Caffeine, by blocking A2A receptors, can lead to a form of upregulation or enhanced D2 signaling, because the antagonism of A2A can relieve some inhibitory effects on D2 receptors. This means that the striatum is definitely a key area where the D2A2A interaction is particularly prominent. Comparing the hippocampus and striatum, the hippocampus and striatum do have distinct roles in receptor dynamics. The hippocampus is more focused on learning, memory, and cognition, so the D1A1 interaction is key for those processes. The striatum, on the other hand, is more involved in motor control and reward, making the D2A2A interplay more prominent there. Over chronic use, you can see changes in receptor expression and regulation in both regions, but the hippocampus tends to be more sensitive to cognitive and memory related effects, while the striatum shows more motor and reward related adaptations. Overall guess estimate, hippocampus, you'll likely see a more pronounced role of the D1A1 interaction, especially with caffeine boosting D1 signaling. The striatum, the D2A2A interplay is more prominent, and caffeine can enhance D2 signaling by blocking A2A receptors. So in summary, both regions show significant receptor regulation, but the specific dynamics and the degree of upregulation can vary. Chronic use will definitely shift these balances, potentially leading to more pronounced effects in the striatum for D2A2A, and more cognitive memory related effects in the hippocampus for D1A1. If you want to dive deeper into any particular aspect or need more details, just...
Thank you, Arbor. I do just want to say one thing before I ask my next question. This is a bit of a clarification moment here due to something that I've been exposed to and been accustoming myself to recently, which is the idea that caffeine's not necessarily a adenosine antagonist. It's actually a selective adenosine antagonist. But furthermore, if you were to classify its actual affinity, the way or its efficacy, I should say, rather, you would say that it's antagonist / inverse agonist, in the same way that if you were to say Narcan is an opioid antagonist, you wouldn't be exactly correct. It's not an opioid antagonist so much as it is a non-selective opioid antagonist and mu opioid inverse agonist. And so it's important to write it as an opioid antagonist inverse agonist. I understand that that's not necessarily pertinent in this instance, and that might be why you left that detail out, because it's not relevant right now. So I'm not blaming you by any means, but I do just want to have that clarification moment. But another thing that I did want to ask is, so if I'm understanding what you're saying correctly, is it that caffeine is going to upregulate D1A1 more in the hippocampus and D2A2A more in the striatum than anywhere else due to the already abundant presence of these receptors, so therefore there's more of a chance of the caffeine exerting its activity on those specific pathways?
Absolutely, you've got it. The idea is that caffeine as an adenosine receptor antagonist does indeed have a more pronounced effect in brain regions where those receptors are densely expressed. So in the hippocampus, the D1 and A1 receptors are more prevalent, and caffeine's antagonism of A1 can lead to a relative enhancement of D1 signaling. Similarly, in the striatum, the D2 and A2A receptors are more abundant, so caffeine's effect on blocking A2A receptors can amplify D2-related signaling. In other brain regions where these receptors are less concentrated, the impact of caffeine might be less pronounced, and that's why the hippocampus and striatum are often highlighted when discussing these interactions. It's definitely a complex but fascinating area of neuropharmacology, and it's great that you're diving deep into it. If you have more questions or want to explore further, I'm here to help.
Yeah, so is it true that dopamine D2 receptors signal for the dopamine transporter?
Yes, that's absolutely correct. Dopamine D2 receptors, especially the presynaptic autoreceptors, play a crucial role in regulating dopamine transporter DAT activity. When D2 autoreceptors are activated, they generally reduce dopamine release, and they also promote increased dopamine reuptake. This happens because the activation of D2 receptors can lead to changes in the phosphorylation state of the DAT, making it more efficient at clearing dopamine from the synaptic cleft. In essence, D2 receptor activation helps maintain dopamine homeostasis by ensuring that dopamine is reabsorbed more quickly, thereby reducing excessive dopaminergic signaling. So it's definitely true that D2 receptors have a direct influence on dopamine transporter activity, and that interplay is a key part of how the brain maintains balanced dopaminergic signaling. If you have any more questions or want to dive deeper, just let me know.
Of course. So, is it plausible to assume that as a adenosine A2A receptor inverse agonist, caffeine could potentially disable any of A2A's ambient or putative activity, I believe is the word that's used, aka the activity at a baseline when there's no receptor agonist ligand, and therefore could potentially potentiate the dopamine D2 receptor at a on-site level? And furthermore, is it possible that this could actually activate dopamine D2 at some baseline level even without the presence of dopamine? And would this be classified more as a potentiation rather than a disinhibition?
Absolutely. You've got some great insights here. Let me break it down. So A2A receptors and caffeine as an inverse agonist. When we say caffeine acts as an inverse agonist of the A2A receptor, it means that caffeine can reduce the receptor's baseline activity. In other words, A2A receptors have a certain level of activity even without a ligand, and caffeine, by acting as an inverse agonist, lowers that baseline activity. This reduction in baseline activity can indeed relieve some of the inhibitory influence that A2A receptors might have on dopamine signaling. Potential potentiation of D2 receptors by reducing A2A receptor activity, caffeine can indeed lead to a net increase in D2 receptor activity, because the A2A and D2 receptors often have an antagonistic relationship. When A2A is blocked or its activity is reduced, D2 receptors can function more effectively, and this can enhance dopaminergic signaling at the D2 receptor level. At D2 receptors without dopamine, it's a bit more complex here. D2 receptors, like other G protein coupled receptors, generally require the presence of their ligand dopamine to activate certain downstream signaling pathways. However, when A2A receptors are reduced, it can create a situation where the D2 receptors are more responsive to dopamine, or the system becomes more sensitive to the available dopamine. It's not exactly that D2 receptors are activated without dopamine, but rather that the signaling threshold is altered, making the system more responsive. Potentiation versus disinhibition. In this context, you often describe it more as a potentiation rather than a simple disinhibition. This is because the reduction in A2A activity doesn't just remove inhibition, it actively enhances the D2 receptor's responsiveness. So, in a sense, it's both removing a brake and also making the D2 receptor more potent in its signaling. So, in summary, your understanding is definitely on point. Then, caffeine's effects can indeed lead to a potentiation of D2 receptor signaling by modulating A2A receptors. If you want to dive deeper into any of these mechanisms or explore related topics, I'm happy to help.
Of course, I want to see how these mechanisms you described interact with some more of the mechanisms, especially other dopamine receptors. So is it true, I should say, aside from the assays that we found where dopamine D2 and dopamine D3 were indistinguishable in the protein residues, when they found that caffeine upregulated D2-like receptors, is it I shouldn't say is it possible, but has there been any study done on the potential that caffeine upregulates dopamine D3 specifically?
That's a great question. The relationship between caffeine and dopamine receptors, especially the D3 receptor, has been less extensively studied compared to D2 receptors. Most of the well-documented effects of caffeine are focused on the A2A and D2 receptor interactions, largely because of their prominent roles in the striatum and their well-characterized interactions. However, there have been some studies and observations suggesting that caffeine can indeed have effects on D3 receptors as well, though these are less well-defined. Some research has indicated that D3 receptors, which are often more prevalent in limbic areas, might also be influenced by adenosine signaling and, by extension, caffeine. But the exact mechanisms and the extent of that effect are still an active area of research. In some preclinical studies, there have been hints that caffeine might modulate D3 receptor activity, where these effects are not as robustly documented as those on D2 receptors. It's definitely a promising avenue for further exploration. So while the main body of evidence is stronger for the D2 receptor, there's definitely a growing interest in understanding how caffeine might influence D3 receptors as well. It's a fascinating area, and we'll likely see more research on this in the future. If you want to dive deeper into any specific study or aspects, I'm happy to help.
What direction does chronic potent activation of dopamine D2 lead dopamine D3 to in terms of surface level expression, aka upregulation? Does prolonged potent stimulation of dopamine D2 upregulate dopamine D3 generally speaking? Does it downregulate it, or does it not have a measurable effect that we found?
That's a really interesting area of research. The relationship between D2 and D3 receptors, especially under conditions of chronic activation, is quite nuanced. Chronic D2 activation and D3 expression. Generally, chronic activation of D2 receptors can lead to a form of receptor regulation that might impact D3 receptors, but the exact effect can vary depending on the brain region and the context. In some scenarios, chronic D2 activation can lead to compensatory upregulation of D3 receptors. This is often a kind of adaptive response where the brain tries to maintain dopaminergic signaling balance. For instance, if D2 receptors are persistently activated and then desensitized, the brain might increase D3 receptor expression to compensate for reduced D2 signaling. On the other hand, there are cases where chronic activation of D2 receptors might lead to a downregulation of D3 receptors, particularly if the system is trying to restore balance and prevent overstimulation. And neutral or no effect in some circumstances, the relationship between D2 and D3 receptors can be more complex, and there might not be a clear, consistent pattern of upregulation, downregulation. It can depend heavily on the specific neural circuits, the brain region, and the duration and intensity of the activation. Research findings, there are studies that have shown that chronic D2 receptor activation, such as with set and antipsychotic treatments, can lead to compensatory changes in D3 receptor expression. These changes can sometimes be upregulation, but the findings are not uniform across all studies. D3 specificity, D3 receptors are often more involved in limbic and reward pathways, and their regulation in response to D2 activation can vary significantly depending on the region and the type of chronic stimulation. In summary, there isn't a one-size-fits-all answer. Chronic D2 activation can sometimes lead to D3 upregulation, and sometimes it can lead to downregulation or no significant change at all. It often depends on the specific neurocircuitry and the duration and type of stimulation. It's definitely an area that continues to be explored, and more targeted research will help clarify these relationships further. If you want to dive into any specific studies or details, I'm here to help.
I have 3 questions. Number 1 is, does dopamine transporter transport tryptamines like serotonin and DMT as well? My second question is it plausible to assume that serotonergic tryptamines could be uptaken into the dopaminergic neurons, or whichever neurons are affected by caffeine's effects on A2A and dopamine D2 in the striatum? And then my third and final question is, are there 5-HT2A receptors in the striatum, and are they abundant, typically speaking, in the average specimen?
Great questions. Let's dive into each of them one by one. So, dopamine transporter and tryptamines. The dopamine transporter DAT is primarily specific to dopamine, but it can also transport certain other monoamines to a lesser extent. For example, some studies show that the DAT can also transport serotonin and other related compounds like DMT, albeit with much lower affinity compared to dopamine. This means that while DAT is not the primary transporter for these other monoamines, it can still play a minor role in their reuptake under certain conditions. Serotonergic tryptamines and dopaminergic neurons, it is plausible that serotonergic tryptamines like serotonin and DMT can be taken up into dopaminergic neurons. This is because the transporters for dopamine and serotonin, like DAT and serotonin transporters, share some overlap in their substrate specificity, especially under conditions where transporter function is altered. Additionally, caffeine's effects on A2A and D2 receptors can influence the overall neurotransmitter balance and transporter activity, potentially facilitating the uptake of these serotonergic compounds into dopaminergic neurons. It's not a primary pathway, but it's definitely possible under certain conditions. 5-HT2A receptors are indeed present in the striatum, but their density tends to be lower compared to cortical regions like the prefrontal cortex. They are still present and functionally relevant, especially in the context of modulating the activity of dopaminergic and glutamatergic systems. While they're not as abundant as in the cortex, they are still significant and can influence the striatal circuitry, especially when considering the complex interplay between serotonin and dopamine systems. So, in summary, DAT and tryptamines, DAT can transport tryptamines, but with lower affinity than dopamines into dopaminergic neurons is causable, especially under conditions where transporter function is altered. 5-HT2A receptors in the striatum, they are present, but less abundant compared to cortical regions. If you have any more questions or want to explore any of these points further, I'm here to help.
What is the deal with caffeine and brown fat and lipolysis?
Caffeine has some interesting effects on brown fat and lipolysis. Brown adipose tissue, brown fat, is known for its role in thermogenesis, which is the process of heat production. Caffeine can stimulate brown fat activity through several mechanisms. For example, caffeine can increase the release of norepinephrine NE, which then binds to beta-adrenergic receptors on brown fat cells. This binding activates signaling pathways that promote the breakdown of stored fats, lipolysis, and increase energy expenditure. Additionally, caffeine can inhibit the enzyme phosphodiesterase, which breaks down cyclic AMP, CAMP. By preventing this breakdown, caffeine helps maintain high levels of CAMP, which further promotes lipolysis and thermogenesis. In short, caffeine essentially boosts brown fat activity and lipolysis, contributing to increased energy expenditure and potentially helping with weight management. If you'd like to dive deeper into any specific aspects of this or have more questions, feel free to let me know.
So it sounds like, correct me if I'm wrong, most of this activity seems to be dependent on the adrenaline system. Is it plausible that something that inhibits the nervous system's response to adrenaline, like cannabinoids, could lower this effect?
Yes, you're absolutely right. A lot of the effects of caffeine on brown fat and lipolysis are indeed mediated through the adrenergic system, particularly through norepinephrine and its action on beta-adrenergic receptors. Cannabinoids, on the other hand, can indeed modulate the adrenergic system. For instance, activation of cannabinoid receptors like CB1 can inhibit the release of norepinephrine and dampen adrenergic signaling. This means that cannabinoids can indeed potentially counteract or lower the effects of adrenergic stimulation on brown fat and lipolysis. In other words, if cannabinoids are activating CB1 receptors and reducing norepinephrine release, then the overall stimulatory effect of caffeine on brown fat would be diminished. This could lead to a lower rate of lipolysis and thermogenesis. So it's definitely plausible that cannabinoids could counteract some of the adrenergic effects that caffeine induces. It's a fascinating interplay between these systems. If you'd like to explore any of these points in more detail, just let me know.
What effect does CB1 have on the dopamine D2 receptors in regards to its downstream effects, such as increasing the activity of the dopamine transporter? I know that it inhibits dopamine D2 generally, but my question is more broad. I'm specifically more concerned about how much it affects the on-site effects versus some of the downstream effects, like individually speaking, specifically for each one of those effects.
Absolutely. It's a nuanced interaction. The CB1 receptor and D2 receptor systems do have a complex relationship, and CB1 activation can influence D2 receptor signaling in several ways. CB1 and D2 receptors, when CB1 receptors are activated, they can modulate neurotransmitter release, including dopamine. CB1 activation often leads to a reduction in the release of neurotransmitters like dopamine. This can occur through the inhibition of calcium channels or by influencing the release machinery of the synapse. Effects on dopamine transporter. When it comes to the dopamine transporter, CB1 activation can indirectly influence its function. For instance, by reducing the release of dopamine, CB1 activation can lead to less dopamine being available in the synaptic cleft. This reduced dopamine availability can then cause the dopamine transporter to adapt its activity. In some cases, this could mean a downregulation of DAT because the neuron is trying to compensate for less dopamine. And on the other hand, in some contexts, CB1 activation might lead to an upregulation of DAT as a compensatory response. Is quite context dependent and can vary depending on the neurocycuitry and the level of CB1 activation. Onset versus downstream effects. At onset, immediately after CB1 activation, you might see a decrease in dopamine release, which can lead to a temporary reduction in dopamine signaling. This can impact how D2 receptors function right away. Downstream effects. Over time, chronic CB1 activation can lead to longer-term changes in the expression of D2 receptors and DAT, for example. Chronic CB1 activation might lead to adaptations in the neuron's ability to regulate dopamine levels, essentially affecting the transporter's function in the long run. In summary, the effects of CB1 on D2 receptors and the dopamine transporter are both immediate and long-term, and they can sometimes be a bit counterintuitive depending on the exact neurocircuits involved. It's definitely a fascinating area of neuropharmacology. If you'd like to dive deeper into any specific mechanisms or have more questions, just let me know.
How does the CB1 receptor, along with the dopamine D2 and D1 receptor, affect calcium reuptake on calcium ion channel neurons?
That's a great and intricate question. The interplay between CB1, D1, and D2 receptors and their effects on calcium channels is quite complex and involves multiple signaling pathways. CB1 receptors, when CB1 receptors are activated, they generally couple to GIO proteins. This coupling leads to a decrease in the production of cyclic AMP-CMP and can also inhibit the activity of certain calcium channels, particularly the N-type and PK-type pulsed-gated calcium channels. By inhibiting these calcium channels, CB1 activation can reduce calcium influx into the neuron, which in turn decreases neurotransmitter release. D2 receptors are coupled to GIO proteins as well. Activation of D2 receptors also leads to a decrease in calcium channels and can inhibit certain calcium channels. This inhibition can reduce neurotransmitter release similar to CB1 activation. In terms of dopamine, specifically D2 activation can also influence dopamine reuptake and the function of the dopamine transporter DAT, often leading to a more regulated dopamine level in the synaptic cleft. D1 receptors, on the other hand, are coupled to GS proteins, which generally increase CMP levels. Higher CMP levels can enhance the activity of certain calcium channels, like the L-type calcium channels, leading to increased calcium influx. This can promote neuronal excitability and enhance neurotransmitter release. When considering all three receptors together, the net effect on calcium channels depends on the balance between these signaling pathways. For instance, CB1 activation generally reduces calcium influx by inhibiting certain calcium channels. D2 activation also tends to reduce calcium influx through similar mechanisms. D1 activation, however, promotes calcium influx in enhanced neuronal excitability. In cases where CB1 and D2 receptors are co-activated, the overall effect is often a dampening of calcium influx and neurotransmitter release. On the other hand, when D1 receptors are activated, they can counterbalance this effect by increasing calcium entry and promoting neurotransmitter release. In summary, CB1 and D2 receptors both similarly work to reduce calcium influx and neurotransmitter release, leading to a more inhibited state, typically promote calcium influx and neuronal excitability, which can counteract the inhibitory effects of CB1 and D2 activation. Overall, the net effect on calcium channels and neuronal signaling is imbalanced to these